Now, students, we are going to discuss neurotransmission at cholinergic neurons. This is the figure from Ketzung book. Here it has been represented that we have choline in our extracellular space that is outside the neurons. So, this choline is actually transported into the neuron with the help of a sodium-dependent choline transporter. So, along with the choline, there will be also transfer of sodium into the neuron, and this step is actually blocked by the or inhibited by the drug that is known as hemicholinium. This is the rate limiting step in the formation of acetylcholine. Now, whenever this choline is in the cell, it will react with the acetyl coenzyme A. With the help of the enzyme that is choline acetyl transferase, there will be formation of acetylcholine. So, now we have acetylcholine in the neuron. This acetylcholine is now transported into the vesicle, that is the storage vesicle. This vesicle is present in the neuron, and this is transferred into the vesicle with the help of a vesicle associated transporter, that is VAT. This VAT will transport acetylcholine into the vesicle. As a result, during this process, there will be a flux of a proton, that is hydrogen ion. And this step is inhibited by a drug that is visamicol. Now, in this vesicle, along with the acetylcholine, we also have ATP, that is adenosine triphosphate, and proteoglycan. So, now in this vesicle, we have neurotransmitter acetylcholine and co-transmitter ATP and proteoglycan. Now, whenever there is action potential, whenever there is a stimulus to the neuron, there will be opening of the calcium channels which are present on the neuron. There will be opening of the voltage gated calcium channel on the neurons. As a result of this opening, their calcium will be in the neurons. There will be influx of calcium in the neuron. As a result, this calcium will give a stimulus to this vesicle to diffuse itself, to fuse itself with the presynaptic membrane. Now, this vesicle will fuse itself with this presynaptic membrane. Then there will be a exocytosis process. As a result, there will be expulsion of the acetylcholine along with the co-transmitters, that is ATP and proteoglycan. Now, this content is now in the synaptic space. This process of the release is actually inhibited by a toxin, that is botulinum toxin. This botulinum toxin is a neurotoxic protein that is produced by a bacteria, that is Bacterium Clostridium botulinum. Now, whenever there is release of acetylcholine in this space, this acetylcholine now will bind itself to the receptors which are either present postsynaptically on the cell surface, or this acetylcholine now can bind itself to the presynaptic receptors. These are the autoreceptors which are involved in negative feedback mechanism. This acetylcholine can also bind itself to the heteroreceptors which are present presynaptically. Now, this acetylcholine which is present in the synaptic space can get degraded with the help of an enzyme, that is acetylcholine esterase. So, acetylcholine action is actually terminated by this enzyme, that is acetylcholine esterase, which breaks down acetylcholine into choline and acetate. Now, this choline can again be transported into the neuron, and again there can be formation of acetylcholine. So, when we talk about the vesicle which is present in the neuron that contains acetylcholine, it doesn't contain only one acetylcholine molecules. It contains 1,000 to 50,000 molecules of acetylcholine in the vesicle. So, the vesicles are concentrated on the inner surface of the nerve terminal, and these vesicles always face the synapse. Why they are facing the synapse? Why they are facing the synaptic space? Because of the interaction between the proteins which are present on the vesicle and the protein which are present on the presynaptic membrane, on the inner side of the presynaptic membrane. So, we have protein in this vesicle, we have protein in this membrane. Due to interaction between these proteins, this vesicle is actually spacing or facing the synaptic space. So, These proteins which are present on the vesicle are the VAMP, that are the vesicle associated membrane proteins, and a subgroup of this protein is SNARE, and this protein is actually on the vesicle. Now the protein that is present on the presynaptic membrane, or that is present on the inside of the terminal cell membrane, is actually the SNAP, that is synaptosomal nerve associated protein. Now I have used the word SNARE protein that is present on the vesicle, which is actually a VAMP. SNRA protein is actually a soluble, S for soluble, NSF attachment, A for attachment, R for receptor. So soluble NSF attachment receptor, RE receptor, where NSF is the N-ethyl maleimide sensitive fusion protein, where VAMP stands for vesicle associated membrane protein. So this was all about the transmission or neurotransmission in the cholinergic neurons.